We have invited today to present a project, a therapeutic project on Alzheimer's disease. Dr. Ángel Cebolla, who is a pharmacologist, doctor in pharmacology, who worked during several years, about 10 years as a researcher. Then he set up a small company, a startup, developing a test for disease intolerance disease, and develop a test for the gluten. That was a big success in a company, in the first company. The name was
Biomedal.
Biomedal, the name of this company. And then finally, 5 years ago, 4 or 5 years ago, she has moved to the field of Alzheimer's disease. And in a new company that is named ONEST, they are developing a program to treat Alzheimer's disease with a compound that is an inhibitor of the sulfatases, for the steroid sulfatases. They have demonstrated in several animal and research models that this inhibitor may be useful for treating the Alzheimer's disease. And he was just now to present the findings, the plans, the expectations.
Thank you
for being here.
Thank you. I think that I have my own microphone. Okay, thank you very much for the invitation. In particular to Teodoro Dercer. Dr. Dercer has invited us and has very soon in the development of the company of Olavide Neuron to help us in the design of clinical trials for Alzheimer's disease. Here I will show the role of steroids in a healthy brain, and how we can induce the therapeutic activity in brain by using a drug that can modify the balance of steroids in the brain. So in this center, in this marvelous center, it's difficult to enter in this, to talk about this, because you know very well the problems of neurodegeneration in population. It's the annual cost per patient. The care expenses are 50,000 per patient, so it's very high, and is about 60 million people in the world are affected from some of the neurodegenerative diseases. And there is also the progressive alteration is more and more problematic for the quality of life, and especially for the families and relatives. And the cost is also increasing with the advance of the disease. There is a clear treatment gap. There is no specific treatment that can modify the evolution of the disease so far. We have focused our attention in the steroids, because although everyone has in the mind that they are primarily produced in adrenal cortex, gonads, or placenta, and different stages of the and sex, they can also be produced in the brain. The starting material is cholesterol that can be transferred through different enzymes and to produce the different steroids. There is dozens of different steroids in the body. Pregnenolone sulfate is the sulfated form are very important. You will see later on, and there are two different key enzymes: sulfatase, which produce the sulfatation of the steroids, and sulfotransferase, which add the sulfated to the steroids backbone. So this is another very abundant, the most abundant steroid in the body, in the human body, is the DHEA, dehydroepiandrosterone. So here in the brain, you can see in different regions, you can see the expression of some of these enzymes that are described in this slide, in this figure. And you see most of them or have some of these genes that can be identified there. And as I mentioned before, neurons and glial cells can synthesize neurosteroids. So steroids produced in the brain in neuron and glial cells are called neurosteroids. And maybe the most important, more abundant are pregnenolone, the dehydroepiandrosterone, and allopregnanolone. This one has started an Alzheimer clinical study, phase 1/2. And the other has been already tested with different success rate. One of the most clear molecular signature of the aging is the level of steroids. The concentration of DHEA sulfate is increased to the young adult, and this is the peak for women and men, but it decreased about 1 to 2% per year to the level of 10% or 5% in people older than 70 or 80 years old. So here this a focus in the axis of the sulfatation and the sulfatation of steroids, because free steroids and conjugated steroids are used for direct binding to nuclear receptor and make the activity of the hormones. There is also a downstream conversion of these steroids. And the sulfated form is used for different purposes. It can be excreted in the renal colon, but it can be suppressed, modified downstream conversion, because once you have the sulfate, then there is no change of the steroids to other steroids. There is a direct effect at the level of neurons of the brain, and it can also further metabolize. So in general, this is a more soluble than the free form, and it is also more stable. You see, the DHEA, the free DHEA, have a half-life of half an hour or 2 to 3 hours in the body. The fluctuation is 3 times the level during the day. And DHEAS, the fluctuation in the body is very small, 20%. But the half-life is very high. It's relatively high compared to the unsulfated form, 7 to 20 hours. Pregnenolone is similar. You see, pregnenolone sulfate is more than 4 hours half-life. And this correlates with the abundance in the body. So the sulfated form is like a bank of steroids that are not used, maybe ready for use, but not used. And it should be desulfated to make the hormones action, but as a sulfate, as a sulfated form, it can also act as a neurosteroid. So the concentration in the body is 100-fold or to 1,000-fold more. And pregnenolone, the range is also high, but in older women, post-menopause female, the concentration can be very significantly low. So sulfation could stabilize the level of neurosteroids. And this might be the stabilization could go in the opposite direction that the aging in some way. But this is the idea, one of the ideas that I would like to remark in the speech. So neurosteroids and brain function, the neurosteroids can act directly to several receptors. Some of them are important for the memory and coordination. GABA receptor, NMDA receptor, and also this is in the neuron and in the glial cells. You can also have some targets that are related with the inflammation. So neurosteroids have also anti-inflammatory effect. And you see, you know that one of the key aspects of the Alzheimer's disease is inflammation. So neurosteroids can induce neuronal excitability, can also neuroprotect. Even there has been some description of antidepressant effect, a memory enhancing effect, and it has also some anti-inflammatory effect in the brain. So these are the two major sulfated steroids, and in the literature you can see that the primary neuroactive effects are can act in the beta amyloid toxicity, anti-apoptotic, mitochondrial protection, cognitive enhancement, and anti-inflammatory effect. There are similar overlapping effects with pregnenolone sulfate. One of them is has been not described in DHEAS, is increase in hippocampal acetylcholine release neuroprotection. So it's maintained cholinergic neurons more protected. So the molecular targets are relatively, some of them are different. And the evidence of neurodegeneration model in neurodegeneration model has been the reduction of beta amyloid plaques and enhance of the neuronal viability against anti-amyloid beta amyloid protein. And one thing that has also been observed is that there is a lower levels of sulfated DHEA associated with Alzheimer's disease risk. So it's overlapping a little bit different, maybe because of the study has studied different things, but you can see that maybe each molecule can have a particular activity, enhanced activity for certain functions. In neurodegenerative disease, has been, as I mentioned before, lower plasma DHEAS and reduced sulfotransferase activity are risk factor for Alzheimer's disease. There has been also another study that described 40 metabolites that are modified in Alzheimer's disease compared to healthy population of the same age. And you can see 4 metabolites that are sulfated steroids that are decreased in Alzheimer's disease, 4 out of 14. Parkinson's disease, you can see also alteration in neuroactive steroids level that has been documented. And there is also the ratio is also important because the anticonjugated steroids, more anticonjugated steroids than conjugated steroids, sulfated steroids, are can contribute to pathophysiology, to the pathogenesis. Multiple sclerosis, there are also some anti-inflammatory effects produced for described for DHEAS. So these are some of the papers where you can find some information about that. In Alzheimer's disease, in these regions of the brain, you can see that there has been described some reduction in the amount of neurosteroids. Only one study, it has inconsistent results, but you will see that this is not the sulfated form of the steroids. This is the sulfated, this is the unconjugated form. So most of the steroids are decreased, but consistently, those one with a sulfated form are decreased in Alzheimer's disease. These are the name of the neurosteroids. So neurosteroid action in prevent Alzheimer's disease seems to occur at the level of prevention of apoptosis, oxidative stress, mitochondrial dysfunction, synaptic loss, and regulation of intracellular survival signaling pathways. So as a summary of what we have seen so far, the evidences that has been collected so far before we came to this field is that steroid hormones decrease with age in human, about 2% per year. DHEA is more abundant in long-aged population. You can see that in the Okinawa population, there is a study. You know that there are a lot of centenary people in this island of Japan, and low incidence prevalence of Alzheimer and other neurodegenerative diseases. And the concentration of DHEA is consistently higher than in the United States. I think that it was the reference, the referent data. Calorie restriction and physical exercise increase longevity and decrease age-related neurodegeneration. Maybe these are two of the most efficient intervention for decrease the neurodegenerative disease risk, and some molecular effect that you can see in the calorie restriction of physical exercise is the increase of sulfatation of DHEA. Sulfated steroids, as I mentioned before, is more stable than free form. And the sulfated free steroids decrease this ratio of steroids, sulfated versus free, decrease in Alzheimer's disease. This is a meta-analysis study. As I mentioned before, 4 metabolites decrease in Alzheimer's disease are sulfated steroids. And DHEA, maybe this is a, you know that this is a regulated suppression effect of cortisol. So these act in a like an antidote or something like that to cortisol formation, and this is compromise the survival of new neurons. So you see with the age, DHEA is decreasing to 5%, but cortisol is maintained or even increased. So if this is counteracting the activity of cortisol, there is less and less effect of the DHEA to counteract with cortisol. So we are approaching to our molecule. This is the two enzymes that are related with the sulfatation and desulfatation of the steroids, neurosteroids. Sulfation that increase solubility and creates longer-lived stock pools of steroids. The bank of steroids is increased with the sulfotransferase. You have more stable money for your health in the future with the sulfotransferase activity. And the sulfatation is in the opposite in the opposite direction, but is widely expressed, including brain capillarities, forming blood-brain barrier. So it's modifying even in the blood-brain barrier the steroids. So strictly, this gene, the gene coding this protein, is can be optional in human. You can find mutants of this gene, but this is the way that we discovered this this in our scientists in Olavide Neuron discovered that STS inhibition induced longevity in C. elegans. So it's optional also in C. elegans. If you mutate that gene, then C. elegans increase the lifespan about 10% or 20%. So this is another mutation. And there are some inhibitors of STS that were used in the past for cancer, anti-cancer compound, because STS is overexpressed in cancer cells, in tumor cells related with hormones, for instance, breast cancer. And this is why inhibitor of STS were one strategy for cancer, and that was why it was available to make these experiments, and we can simulate the mutation of the gene in C. elegans. So the group of Manolo Muñoz in Seville, they discovered that with STS64, one of these inhibitor could increase the longevity of C. elegans in a similar way to the mutation. And you can also have a higher level of sulfated steroids in the mutant of STS. So this is demonstrating the action of this gene. So I would like to introduce our candidate for the treatment of neurodegeneration, STS64, also called irosustat. And we try to call it also ONESTX1 because of to make our branding name to add our branding name to the drug. It is a potent inhibitor. Is at that time it was the most efficient inhibitor of the STS, and it has been developed in the clinic to reach a phase 1-2 in different trials for hormone-dependent cancer. So we know from that studies that has a considerable broad wide safety profile, acceptable safety profile. This is the structure. We know also that there are some publication of other STS inhibitor, the DU14, that decrease cognitive deficit in spatial learning and memory, and protected hippocampal synaptic plasticity. So this is a paper describing this the activity of this sulfatase inhibitor, but there were no continuation of this research. We continue with the research of STS64, and this is a zoom of the previous graph, where you can see how the longevity of CLN could be achieved with this with the addition of STX64. But in the mutant of the sulfatase, you cannot have such longevity. So it's due to the inhibition of STX64. These are some of the experiments that was with STX64 and the mutants of STS. One of the model C. elegans model, you can find a lot of models of diseases, or human diseases in C. elegans. And in this model, you can find this is a model of Parkinson's disease with a fusion of alpha-synuclein gene to a reporter gene. And they could see that there was a reduction of the mobility in those with the expression of this synuclein reporter gene, and with the age of the C. elegans, and in a background with the STS minus, you can with STX64 addition, you can find that the mobility, the bends per minute, is the way that the bends is how the worm is moving. So it was improved with addition STS64 or with a mutant. And the dopaminergic neurons were also more resistant to the aging with the addition of STS64. In a Huntington disease model, we could see this is a polyglutamine certified repetition of polyglutamine simulating a Huntington gene in C. elegans. And you could see an improvement of the less aggregation in those background with a mutant, STS mutant, or with a background, the wild type background, but with the STX64. You can see a significant reduction of the aggregates. In Alzheimer's disease model, in C. elegans, you have a model that produces beta amyloid and provokes thermodependent paralysis in young adult stage worm. And the paralysis was ameliorated with the deletion of the STS and with the addition of STX64. C. elegans is too far maybe yet to the human, but when they move, thanks to the group of Dr. Ángel Carrión, they made some experiments in a model of Alzheimer's disease in mice. And you could see that in this passive avoidance test, which is a memory test, that the mice has to remember that there is a problem if they cross certain rooms. And they discovered that the STS64 could recover the memory of the model mice, APPS1. This is a transgenic mice producing beta amyloid. And there were, here you cannot see too much, but there is a reduction also in the aggregation, the black formation of beta amyloids. And here, this is a quantitative study of the beta amyloid area, and you could see a reduction in the frontal cortex and the hippocampus in the mice. So the treatment of in less than 1 month of STX64 partially reverse cognitive deficit and beta amyloid accumulation in the transgenic model of Alzheimer's disease. Well, one of the first sign of neurodegeneration occurring during Alzheimer's disease is the loss of cholinergic functions. So it appeared before some symptoms occur. So you have first an accumulation of protein. This is the first sign, accumulation of beta amyloid. The second aspect that you can see in Alzheimer's disease at the molecular level is the functional cholinergic axon, loss of cholinergic terminal, etc., and atrophy at the very end of the evolution of the disease. So far, you know that the treatment this is the cholinergic neuron producing acetylcholine. And acetylcholine is produced with the last step with the choline acetyltransferase, and they are moved with the vesicles to the synaptic region, and they release acetylcholine. So, and the acetylcholine receptor detect the presence of this acetylcholine. So, so far you know that the only symptomatic efficient way to make some effect in Alzheimer's disease is the anticholinesterase treatment, donepezil, galantamine, or rastimine. And but this is a problem when there is no capacity of produce acetylcholine. So we demonstrated that this mechanism of action of cholinergic activity that produce the sulfated steroids is from the synthesis and release of acetylcholine, because this is a C. elegans with a fusion of choline acetyltransferase and vesicular acetylcholine transporter fused to GFP to fluorescent proteins. And when you measure the fluorescence, you could see an increment with STX64 of the expression of these genes with the reporter. This is the vesicles and different strains. So in mice, you could see also some evidence of the induction of the freshen of the synthesis of acetylcholine, because in medial septum, immunostaining of choline acetyltransferase could be observed. This dot point, black point, and also in the caudopotamen, two regions that are related with learning and memory. You see here the quantitative assay. So this is a clear induction with STS64. And we make an experiment in rats to see if there is a impact of this cholinergic action in the cognitive recovery, in the recovery from amnesia, induced amnesia with scopolamine. Sorry. This is the test that was made. If you add scopolamine, you suppress, you inhibit the muscarinic receptor because it is an antagonist of acetylcholine, and you can have amnesia provoking in rats. This amnesia is measured in a novel object recognition test. This is a test where you enter in a room with two different objects. Well, in this case, the same object, and the time examining the first object and the second object is the latency time. So you have if the rats has a healthy brain, they will recognize the second object as similar to the previous one. So the time of examination is lower. If you don't have such a memory, then you will examine the first as equal to the second one. And this is a new object that is added in the second room some days after the first experiment. So if you have a memory, you will have this shorter time with this one, because the memory, you recognize the object has been explored before, 2 days before, 1 day before, in short-term studies. In amnesia, you will not recognize this one, okay? You will not see this. You will see this object for the first time, and you will have the same latency time for the two objects. So with the scopolamine, that's what you have, the latency, the discrimination index. With the vehicle, with no scopolamine, you have this discrimination index. With scopolamine, you suppress the time for exploration of the two objects. And with donepezil, you have a reduction, significant reduction. I mean, significant, you have an increment with regards to scopolamine. So scopolamine is added before the study, and then you add the rats has been treated before with STX64, 1 milligram of STX64 to 4 milligrams, and donepezil as a control, positive control. And you will see that some part of the memory has been recovered, a similar way to donepezil, but it's a different mechanism of action. So what we expect is we can have added additional contribution if you mix the two drugs, but this is still to be made. So STS64 effectively prevented the scopolamine-induced memory deficits at both evaluated dose, achieving an effect comparable to donepezil, and suggesting that its efficacy may involve modulation of the cholinergic system. And this is the proof that what we are making in the rats is inducing the sulfated neurosteroids. Pregnenolone sulfate is clearly induced in those rats that were treated with 1 milligram or 4 milligram. There is a slight increment with 4 milligrams, but it's not significant because the error bar is this one. So this is what we are modifying there in the rats is the pool of sulfated steroids. So what we know now about the mechanism of action of STS64 is that while we have a clear STS inhibition, very efficient, and this will increase the sulfated steroids. The pool of sulfated steroids change the expression of cholinergic genes and also probably produce other effects that we have to investigate and modulating the action of other functions, for instance, autophagy, because the degradation of the proteins that we observe in animals, and induce acetylcholine production, and this contribute to lifespan extension and neuroprotection. So the conclusion of what we are seeing is that steroid sulfation and the sulfation axis regulates hormone and neurosteroids viability. There is age-related changes, and age is the risk factor for neurodegeneration. And this axis is significantly altered due to aging and in neurodegenerative diseases. There is a promising modulation of this modification with the inhibition of STS, showing some compelling evidence for improving outcomes in Alzheimer's disease and other neurodegenerative diseases. So STS64 represents a promising candidate for the treatment of neurodegenerative conditions. So the strong preclinical data would like to summarize extended lifespan, improved cognition, reduced aggregates, neuroprotection, anti-inflammatory effects observed in I didn't show this data, but at the level of gene expression, you can see some inflammatory genes, pro-inflammatory genes that are suppressed with the action of the STS54 and induce cholinergic activity by inducing the synthesis of acetylcholine. So with the panel of therapeutic solution now for Alzheimer's disease, STS64 or 1STX1 is an oral administration. The safety profile is favorable compared to the last generation of drug with anti-beta-amyloid, and of course can be also affordable. And this disease modification, it's also potential. You cannot have with donepezil other acetylcholinesterase inhibitor. So this justifies that we have started to design a clinical trial to the two major focus will be Huntington disease because of the speed of the to be in the market, and the lower number of patients required to reach the market. Alzheimer's disease is the other one, because we will compete with the acetylcholinesterase inhibitors. So in 6 months we expect to have some data in patients that can be promising, and also we also would like to focus in Parkinson, and also other anti-aging effect that we can have from the addition to humans. So this is the money we need. Well, I would like to thanks to the supporting companies, and also the Minister of Science that has supported part of these studies. The Pablo Olavide, which is the origin, the university where was the origin of this research and inspiring disease. And this is one of our last meeting in winter. Manolo Muñoz is the researcher that discovered the action of this STS in C. elegans. Ángel Carrión is the master of the mouse experiments, the leader of the experiment. And Mercedes was making the PhD with Manolo, was the co-inventor of the action of the application of the STS-54, and the rest were joining to them to bring this new molecule to the market. Thank you.
Any question or suggestion? I have a small question about the effect of the steroids, sulfated steroids. No, sorry, the effect of diet deprivation and exercise on the level of sulfated steroids. Do you have any idea or any hint about what would be the cause, the reason for this effect?
Well, we have some working hypotheses about that. One of them is that I think that there is, I mean, probably this mechanism of action is related to the metabolism, and it seems that sulfotransferase is induced in C. elegans with the diet, I mean, with the fasting. So the scarce, if you have a lack of resources for eating, for having the energy, you can have this activation of the sulfotransferase. So we think that this has been also conserved in human or in mice, this sulfotransferase induction, because I mean, once we have this hypothesis, I check in the literature if there were some documentation about that, and there were some papers indicating that after physical exercise, you have a induction of the sulfatation of the DHEA. And there is also a consistent result that DHEA is more abundant in people having regular exercise. So it has some relationship with the metabolism. Maybe mitochondria is also involved in that, the protection of the mitochondria, and autophagy. Maybe when you are lack of energy, you take what the cells have, take anything that can found in the cells as a waste. For instance, aggregated proteins. Aggregated proteins can be, maybe it are mobilized, are disaggregate partially to have more resources for maintaining the function in fasting condition or in conditions where you have some need extra energy.
If I understood correctly, these sulfated neurosteroids increase resilience of the brain, but we lose when we age part of this capacity, and we especially lose when we have neurodegenerative disease, Alzheimer's disease. Do you know why neurodegenerative disease is affecting this phenomenon?
In this case, you have an egg and a chicken, and what is first the chicken? In this case, I think that if you have a decrease of the neurosteroids, then you have more risk of neurodegeneration. So what it came first is the decrease of steroids, and then you have less protection to the brain. So probably there is then a feedback negative loop, maybe, and you have more destruction of capacity to maintain the level of steroids if you have the neuro destruction of neurons. So I mean, you can accelerate that, maybe that process. But I think that the lower level you have more risk for neurodegeneration. Probably it came first, the lower level of neurosteroids.
But of course, could be a pleiotropic effect that the thing that is causing the decrease of these sulfated neurosteroids is also causing Alzheimer's disease. Because in Alzheimer's disease, we know that lipid it's one of the risk factors. So we know ApoE4 is very important. It's the most important gene, and it is a lipid transporter gene. And there are many lipid genes that are detected in the GWAS as a risk for Alzheimer's disease. So this could be kind of that they are maybe not that related, but they have a common factor that kind of statistically makes this. This could be a possibility.
Completely feasible. I think that is an interplay of maybe of different. Well, we know that there is accumulation in the liver, in the rats when you add STX 64. So somehow with these steroids, we are moving metabolism in some direction. There is a decrease also, partial decrease of glucose, increment probably of some lipids. Probably it's there are no radical changes, but continuous changes that are long term you have consequences probably.
So, for example, the studies that you were mentioning, that there were difference between Alzheimer and controls in terms of these lipids and these neurosteroids. I wonder whether they took in consideration ApoE4, because of course you have more ApoE4 in Alzheimer's disease, and this could also influence the levels of these neurosteroids. ApoE4 is the risk allele of Alzheimer's disease, and it's also very important for lipid transport. So I guess this is something that.
Maybe of course this is very complex, because the as you see the neurosteroids act maybe not modifying radical changes, but subtle changes in many parts, and maybe there is a coordination. Lipids probably there are many markers, many genetic markers related with the lipid metabolism. So probably it's also somehow linked.
In any case, I think in your trial you should take into consideration ApoE4, because I think it could be an important modifying factor. Of the effect.
We will take into.
Thank you. Just a curiosity, the curve that you showed during the lifespan of the individual of the level of sulfated neurosteroids, well, it somewhat reminded me to the curve of adult neurogenesis. Do you know if there is any relationship between these compounds and neurogenesis?
There is we have some suspect of neurogenesis. I didn't show the slides, but there is a more density of cells after treatment with STX 64.
Mhm.
So there
very
is we suspect that, but this is, I mean, this is a big thing.
Yeah,
So we
exactly.
need to be very careful with that. So I mean, the repetition several times and different people. So, but I mean, what we have so far are promising,
Okay.
and we have seen also some evidences with neurosteroids.
Yeah.
There are some publication about that. So we
So-
we are optimistic in that we can have certain neurogenesis.
Okay, thank you.
Hello. So my question was about the levels of the neurosteroids that you were mentioning. So obviously we have seen that if the bank that you mentioned, that reserve, falls below a certain point, it increases the potential for neurodegenerative diseases to advance. But is there a point where there's too much reserve and that can also cause problems?
That's a good question. I think that there is some kind of homeostasis in the body preventing that. I think if you don't, I mean, if you have suppression of STS, then probably you can accumulate neurosteroids if you add new steroids. But what we are modifying are the endogenous steroids, and they are not. I mean, I mean, we know that there is a direction to decrease neurosteroids. So if we in the ideal situation we maintain them, but to increase, maybe we can have in a pathological situation that's doping, you know, and especially for young people. Maybe for older people there is no problem of adding steroids. There are not too many studies about that. So steroids is a kind of taboo, because of the bad press about to have steroids, you know, in the gym, in the forbidden. Even they have Manolo has problem to get steroids to make experiments because he had to sign 20 papers to have some asteroids. So there is a bad press, but the utilization for old people, I think that it has never been seriously considered. I think, and there are few publications about that. So you unfortunately, people are even in the pharmaceutical companies. They are focused on the people from mid age to the childhood, so to the babies, but not for the old people. Maybe the old people is a new league. I mean, you have to maybe you can have a new rules maybe for even for the treatments. And but this has to be made because you know sarcopenia. There are some problems osteopenia. There are some problems that also link, and maybe can be recovered with steroids. I don't know, but this is I mean this is a bad press and maybe a taboo talking about steroids in general.
Okay. So just to do a little follow-up question on that. When I first asked the question, you mentioned that you see a difference in levels of sulfated versus non-sulfated, but not an increase in the overall amount of steroids. Is that correct?
You're right.
Okay. So
well, in this case, in the rats experiment, we see a higher level of sulfated steroid, but the previous studies in the XTS64, we see an clear increment of the ratio, but not the
overall.
Not very significant the whole level of steroids. Yeah.
Okay. So my follow-up question to that would be then: is it possible that instead of the sulfated versions being neuroprotective, the non-sulfated versions are pathogenic?
It's feasible, yeah. It's another possibility. Yeah, completely.
Good.
Right. In fact, there is one publication in a postmortem study in brain of Alzheimer that the DHEA was increased in Alzheimer's disease, but the sulfated was decreased.
Okay.
This is the only study. I don't believe too much in a single study. I would like to see several evidences in different laboratories.
Okay, well, thank you very much for the presentation. That was very interesting.
You're welcome. I want nice talk. Yeah, thank you. Have you or has anyone tried STS-64 in ALS? Not so far, but we are looking forward to use it in ALS because I think that it's one of the target. I mean, one of the disease that has been some proof of concept in animals with neurosteroids. So if everything that works fine with neurosteroids should work with STS-64. So, but our money and resources don't have enough energy to start a project in ALS, but we are willing to join any group that would like to collaborate with us in that sense.
Any other question? In relation to the last question.
Charo Osta, thank you. Thank you to you.
Yeah. Relation with the last question about the ALS, you have presented several data about the positive effects of the level of steroids, especially of sulfated steroids, on the several aspects of cell homeostasis and cell biology, especially of neural cells. And so you have more or less concluded or suggested that the treatment with the inhibition of the sulfatase, steroid sulfatase, could be effective or positive in several neurodegenerative diseases. If
yes.
Not in all neurodegenerative diseases.
Well, like physical exercise is like that.
That's true. Physical exercise is positive in many, but what would be, in your opinion, the optimal target, the best
target?
Target, where we could expect the best results.
So far, we only have a four model system: Parkinson, Huntington, and Alzheimer. We have more evidences for Alzheimer because we have this cholinergic activity, what is relatively specific of Alzheimer's disease, and also Lewy body dementia. I think that these are the two major neurodegenerative disease because we discovered this action of related with the cholinergic function. Maybe, but the other were not, the results were not so spectacular as we see in memory recovery.
So we can expect that your compound will show efficacy firstly in Alzheimer's disease.
I think that well, so far we have more evidence, preclinical evidences in Alzheimer's disease. Yeah.
Well, everybody were results.
Yeah, in fact, this was our first focus. But well, Alzheimer's disease didn't like too much to venture capital because it's it's risky because there are a lot of failures even in phase 3. It's expensive because you need a lot of patience and a long time for having results. We try to focus on symptoms to make it shorter. Well, these are the three major problems we have with venture capital, and the other one is that a repositioning drug is not a new entity, but this is as I told you many times, this is a less risk because it has been already used in human, and we have this. We know that this is not. It's safe in human. We hope that we will have some contribution to Alzheimer's disease with that.
Yeah, expect good luck for you.
Thank you.
Thank you.
Thank you. Thank you.